Hello, everyone. Thank you so much for joining today. I'd like to begin with a recap from our presentation, last month. Last month, we were talking about why brighter is not always better. And it's not always better because it doesn't take into account spread. So what we're looking at here is a single color control stained with PE E-fluor 610. From the plot on the left, you can see that we have some spillover. We have a double positive population with a single color control. So we know something needs to change, and that change is compensation. When we compensate, in other words, the MFI, or the fluorescence intensity of the PE E-fluor 610 in the blue laser 2 channel, is reduced and normalized to the unstained population, you'll see that we have this spread. We're still observing cells that appear to be double positive. This is because it's just an inherent quality of the dye, PE E-fluor 610. In actuality, the compensation process, it calculates and adjusts all of the emissions of PE E-fluor 610 into secondary detectors, and it adjusts it to practically zero. These are normalized means, but it does not take into consideration the spread of the dye. So although it seems exaggerated by compensation, the spread, it can't be changed through compensation. So what do we do with compensation? So how do we recognize spillover and compensation? The best way to recognize it is to actually run your compensation samples as samples in the experiment so that you have more functionality in the software to look at the data. With the compensation off, a single color control for Alexa-4 488, you can see that it's spilling a little bit into the blue laser 2 detector. What this means is that these cells in right here are false positives, false PE positives. And you can see that on a histogram as well. Here you can see a second peak. On the second peak are the events that are in this double positive quadrant. Now this is only seen with Alexa-4 488, so we shouldn't have any double positives. When we perform compensation, you'll see that the fluorescence intensity of this population has been lowered to be equal with the unstained cells. As a result, the second peak is no longer there, so we no longer have false positives in our data set. So why do we need to compensate? We need to compensate because each dye has a spectral properties and an emissions spectra. This is area of green is what we call the emissions spectra, and in this case, it's for Alexa-4 488. And the yellow peak is PE. The shaded areas are the bandpass filters that are used to collect the data. The problem that we are observing right here is spillover of Alexa-4 488 into the PE channel. These are the false positives I was speaking of before. When there are more than two fluorophores in an experiment, it's important to check on the spectra viewer whether or not you're going to have areas of spillover and thus have to perform compensation. As you can imagine, as your experiment becomes more complex, Here we have an experiment labeled with several fluorescent dyes, and as a result, there are many areas of spectral overlap. All of these areas will need to be subtracted from the data set, remove the spillover, in other words, so that you can analyze the data and that your data is accurate with no false positives. So how do we do this? Well, it's basically done through single color controls. And it's so important. We can't overemphasize how important it is to have good single color controls in order for your compensation to be accurate. So let's talk about whether or not to use cells or beads for compensation, an antibody capture bead. You want to use cells, but those cells, if you want to use cells rather, then those cells have to be the same cells that you are using in your samples. So that the autofluorescence matches and other properties as well. However, sometimes when an antigen is expressed at a very low level, it's difficult to get a bright single color control from cells. And the way to approach that is to use an antibody capture bead, which I'll talk about in just a moment. Now, if you're using any functional dyes, such as cell tracking dyes, proliferation dyes, apoptosis dyes, if you're using those, then there is no choice. You must use cells to create the single color control. And sometimes, but not, it's pretty rare, but sometimes cells can stain more brightly than labeled compensation beads. I have observed this with MHC class 2 antibodies, in which the labeled compensation beads were dimmer than the actual cells. And so this actually would lead to a situation of undercompensation. So let's talk about antibody capture beads. So big advantage, for example, with low expressing antigens, is that labeling the bead will give you lots of positive events. They provide a bright signal. And this is very useful when the expression is dim or the cells are very rare. So, and again, that's where I come to this, where compensation beads, when they can substitute for cells, save your cells for the experimental samples. So if you have a precious type of sample, precious hard to obtain sample, I'll put it that way, then your cell number is usually pretty limited and you don't want to waste cells with creating controls. And so antibody capture beads are definitely the way to go outside of the situations that I've spoken of before. So how will I know if my single color control is good? And by good, I mean good enough to give you accurate compensation. So each single color control is you need one for every fluorophore in the experiment. That includes functional dyes and viability dyes. There's basically like 5 conditions that you would like your single color control to meet. The first condition is that it must be as bright as the experimental samples. If you run your full stain sample first and note the position of the peaks, and then look at your single color control, if the single color control is not equal, as bright or brighter than your sample, then some tweaking needs to be done. That single color control could lead to an undercompensated sample or an undercompensated channel or detector. Consideration number 2 is that if you're creating single color controls, they must match spectrally to the experiment. For example, you're working with cells and the entire population of the cells are GFP labeled or GFP positive, okay? You can't label beads or other cells with FITC or Alexa 488 for your single color control. The emission spectra for FITC and Alexa 488 are very different from GFP. So if this is a case where you do not or are not able to provide a single color control for your GFP labeled cells, we do offer on the Thermo website a GFP labeled bead. It's pre-prepared, and we would encourage you to try that, as well as some of the other fluorescent protein beads that we have. The third consideration is that the unstained and single stained events must match for autofluorescence. For example, if you are using labeled beads to compensate, then the negative beads, the unstained beads, must be used to calculate the compensation. Unstained cells may have a very different autofluorescence level than your labeled beads. So it's important that that these be equal in autofluorescence for the compensation to be accurate. Fourth consideration, if you're creating a single color control using a tandem dye, like PE Sci 5 or ABC Sci 7, you must prepare the single color control with the same lot and preferably the same vial that you have used, antibody vial that you have used to stain your cells. The reason is that tandem dyes are susceptible to degradation each time they're removed from the refrigerator or exposed to light. And so you want those conditions to be mimicked in your single color controls. The fifth condition, single stain control should be brighter than any spillover. As we've shown in our previous slides, the FITC and the PE signal, where we saw the false positives in the PE channel, if you see something like this when you run your single color controls, then you need to take another look at them. So what I usually recommend is to run your single color controls, looking at that control in every other channel you're using in the experiment. If you see that your single color control is brighter in another channel, not the target channel, it's still that dye, but you need to, it's going to be overcompensated, in other words. And so you will need to make some adjustments to your compensation controls to make sure that the single color control is brightest of all the other spillover channels. And so you'll have accurate compensation. So the key takeaways from this session would be to encourage you to design a panel with an effort to keep compensation to a minimum. We recommend that you use a spectra viewer to get a preview of the emissions spectra of your, say, your whole panel, so that you can see those areas of overlap and basically know where those compensation values are going to be important. You want to ensure that your single stain controls meet all the best practice criteria that I just outlined. And avoid adjusting compensation by eye. When we compensate by eye, we almost always overcompensate. So if you see errors, revisit the data. Start looking at it in many different ways, and maybe you'll get a clue as to what has happened. So just in summary, we have all of these different situations that might occur when looking at your data set. So this is a plot. We have CD3 versus CD4, and this is uncompensated data. So we can see this population is in the double positive quadrant when it should be in the single positive quadrant. We apply some compensation, but not quite enough. you can see that it's still a portion of your population is in the double positive quadrant. In the third plot, we see correctly compensated data. The fluorescence intensity of your stained cells is normalized to the signal intensity of the negative population. Here we see a slightly overcompensated sample. And if you were to compare the median fluorescence intensity of this population to the median fluorescence intensity of the unstained, you would see that this one had a lower fluorescence intensity. It's a little overcompensated. And then there are severely compensated, overcompensated samples sometimes. Again, compensation would need to be adjusted to bring this population, the median fluorescence intensity of this population, into parity with the unstained samples. So in closing, I would like to thank you for participating in this talk, this event, and would be happy to take your questions.